No, just pretty much been doing section bank and then have more time now because I'm almost done with classes. So
Got it.
um
All right. What would you like to go over today?
I guess before the section bank, I was just having some trouble understanding Zeff and like ionization energy. Um I think the part specifically about like electronic repulsion was kind of confusing me. I think last time that we had talked, and then maybe just go into some section bank questions. I've been trying out like mapping, and I feel like that's been keeping me actively engaged with the passage and making it a little bit easier, but I still feel like I need some practice with it. I don't know if when you're doing the section or you're doing any passage, are you like mapping out what you're doing, writing out what you're doing to kind of,
So I could show you what you can do for the science passages. It's not exactly like a map like in cars, uh, but yeah, I can kind of show you.
just trying to pull up what, yeah, show you. I don't know if this is something like, um, maybe we don't have to go over this passage today because,
That's good. That what you're doing is good. That's like what I was going to show you.
okay, okay,
Like,
okay,
perfect. The only thing I would change is that the proportionality relationships, like for instance, this one, you would write it like proportional, or if it's an inverse proportion, it'll be like inversely proportional to G-actin. And if it's proportional, it'll just be proportional. So that way you can answer questions about how a graph of it would look like.
okay.
Because they could ask you like, what would a graph of this look like? Um, so doing it like this is the only thing that I would say to do. But mostly everything else is pretty much exactly what I would, you know, how I would like handle it. So good job.
Um, okay. Let me see if I can change let me bring this over here. Can we, maybe we can just go through one of the passages that, ooh, got a lot wrong there. Okay. Um, um, so that one we just did. No. Sorry, I'm just having some trouble
Mhm.
with, why it jumps to command. Okay. All right. Maybe if you have like how you would kind of like map this one out, what would be your, like, I mapped it out and like, I don't know, mine, well, this is not gonna help showing it on the screen, but mine is just like crazy and like I mapped it out and I didn't even like refer back to it um
Uh-huh.
for this one. So
Okay. So, all right, let's see. Um, so yeah, for the science passages, you want to look out for something called buzzwords, which are just words that you're expected to know that they could ask you questions about. So I'll read this out loud and I will tell you um like the buzzwords and stuff. So, okay. HIV integrase, so IN, and it's an enzyme because of the ASE ending. And you'll see a lot of passages about enzymes, and then they'll tell you something about the enzyme's active site containing some type of like amino acids that have some special characteristics, like, either they're charged or either they can form hydrogen bonds and stuff. And usually what they do is they can make like an experiment where where they mutate amino acids in the active site of an enzyme with like alanine, and then they can kind of measure the kinetics and see if, like, things change. So that's just something that you'll see a lot. So okay, HIV-1 integrase is a viral enzyme that mediates the introduction of viral DNA, vDNA, into the host chromosome. So IN just helps DNA from the virus, from the HIV virus, enter into the host cell's DNA or chromosome. Okay. IN is a 288 residue protein. So when they say that, you want to be, or that's like a buzzword because they could ask you the size or the weight of this enzyme. And that's where you would have to know that uh, the average weight of an amino acid is 110 Daltons. So, they'll likely ask you a question about that. But okay. Um, Composed of three domains, the N terminal domain, that's just, the N portion, like side of it, because when you have an amino acid, you have an N terminal, you have a C terminal, and if you make, a big peptide, you're still going to have the N part and the C part, the C terminal. So you have those, but you also have this catalytic core domain, CCD, and that's going to be, the important one where, aka the active site. So, okay, the CCD contains three highly conserved residues, D64, D116, and E152. What amino acids are those?
Is that a DE? So is that no, is that aspartic acid? DE. No, it's
It is.
okay. Yeah, spark. So D and E, right?
And
I think
then,
yes. Are those the acidic? There's KRH
uh,
and there's D and E. So the D's are the I think okay. I think they're the basic, right? The
no,
basic.
the acidic ones. So you have
Oh,
a
okay.
D is aspartic acid and E is glutamic.
Glutamic acid. That's right.
So yeah, so that's what we're used to, like what we should expect to see in the active site of an enzyme, something like an amino acids that have something special to do with them, like charged or hydrogen bonds or something like that. So in this case, it's the charge because they're negatively charged. So that should, oh, and then they even tell us afterwards. So, and mutation of any of these three residues abolishes IN activity. So, a lot of times they'll say that in the form of an experiment, but that's something that we're likely going to see a lot of. So, okay. Now, next paragraph, integration of vDNA in the host chromosome proceeds by two well-characterized steps, both of which are catalyzed by IN. In the host cell cytoplasm, each end of a newly synthesized vDNA segment is bound at a specific long terminal repeat LTR nucleotide sequence from one IN dimer. Okay, so I'm gonna like try to visualize this to understand what's happening here because okay, so let's see. We have of each end of a newly synthesized. So I guess this is like when it's already inside of the host cell. So, oh, so okay. They said that it helps integrate viral DNA into the host chromosome. So it is already inside of the cell. It just has to put itself into the, I guess, DNA of the host cell, but it's already in the host cell cytoplasm. So, okay. So each end of a newly synthesized vDNA segment is bound at a specific LTR by one IN dimer. So I'm going to draw like, let's say this is the vDNA, and each end has LTRs, and at each end, they are bound to an IN dimer. So IN, IN, then here as well. Oh, and they might ask you, like, what's the weight of an IN dimer, which is just, we do what we did with the 288 residues and just multiply it by 2. Okay. Upon binding, IN catalyzes cleavage of a GT dinucleotide from each 3' vDNA terminus. Okay. Still bound to either end of the vDNA segment, the IN dimers join to form a tetramer, which then binds host cell lead GF at the CCD. Okay. So I think these join up. So you can have IN, 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 like joining up at the IN dimers. So, and then this is like the vDNA. And yeah, and they could ask you about the weight of the tetramer, which is just going to be what we talked about before, but multiplied by 4. But okay, so once this happens, after the tetramer forms, it binds to host cell protein LEGF at the cDNA. So maybe something like this can bind to it. And when this happens, let's see, this complex enters into the nucleus, and when it's in the nucleus, the LEGF can bind. So like, let's say this is like a chromosome, the LEGF, I'll just write it as L, binds to it. Well, chromatin, so that's, before the like the chromosome is when it's condensed, so it's not really the way I drew it here. But I'm just trying to show that it binds to the DNA. And then it allows like the vDNA to enter into host cell DNA. So that's how I would do that. Like I would just do that to help me visualize it. Okay. And then they say small molecule IN inhibitors show promise as antiviral drugs against HIV-1. One class of these inhibitors are legends, which bind IN and shift its oligomerization equilibrium toward an inactive tetramer that cannot bind vDNA. Um, so legend could be inversely proportional to, let's say, IN like activity, because it says that it causes IN to form an inactive tetramer that cannot bind to vDNA. So I wrote it as an inverse proportion. And that just means if legend goes up, ion activity goes down. Okay, and then they said another class of small molecule inhibitors are short length oligodeoxynucleotides, ODNs, whose sequences mimic LTRs. Okay, so they look like the substrate, so they're going to be competitive. Oh, and they say it afterwards too. ODNs are competitive inhibitors of vDNA for binding to IN. So ODN is going to be inhibiting the, I guess I could also say like IN, but maybe I'll just write like competitive. So I think that's what I would draw or write out for all of this.
And what was your reasoning? The reason why you don't do the air up and down because you want it in the graph. You can interpret into the graphical. What was your reasoning again? Why you prefer this as opposed to the arrows up and down?
Yeah. So mathematically, so if they asked you, like, okay, let's say we had A is proportional to B and we graphed this, this would just be like that, right?
Hmm.
And or let's say that we did it like this. A goes up, B goes down. What would a graph of that look like?
Hmm. Would it be like kind of the kind of like that? I don't know if you can see my yeah.
So that's what it might appear to be, but let's try this way. So let's say, okay, if A is 1, 1 over B is what?
One.
If A is 2,
0.5.
1 half, right? Yep.
Yeah.
A is 3.
Point three or one third, sorry.
If A is 400,
Uh, 1/400.
so this, as A increases, B decreases, but what number can B not be?
Negative or zero?
0, right?
Yeah.
That would be 1 over 0 undefined. So that really means that this B value gets infinitely smaller as A increases, but it'll never touch 0. So these axes are asymptotes, and the graph actually looks like this,
Okay.
meaning it's like a hyperbolic, and it
Okay.
never touches the axes. So yeah, so like, I always see people do it this way, like including most other tutors. And I actually made like a couple of months ago, maybe like a short video where I'm like, don't do this, but that's, doing it like this with the arrows, like that doesn't give you any insight about like the mathematic proportion between something, right? Because also they can ask you a question like, like if FG is G M1 M2 over R squared, and they ask you, okay, what happens to the force if you double the radius? What would happen?
It quadruples. Like,
So we see that there's an inverse square proportion between the force of gravity and the radius, right? So yeah, so that's why you want to put it in like the proper proportionality terms, because not only could they ask you about, what a graph of an inverse proportion looks like, they could ask you like, in questions, they could ask you, things like what is this proportional to, or what is this, inversely proportional to? And with that, know how it would look mathematically.
got it.
Mhm. In Psych Soc, that's the only place where it'll be different because they use positive correlation and negative correlation instead of directly proportional and inversely proportional. But that's the only one that'll be different. Yeah.
Okay. Okay. Oh
Mhm.
yeah, I forgot that they'll use graphs too, I guess, in Cycloish. Yeah. Okay.
So, so yeah, like any questions about how I went about this?
No, I like it. I mean, I like how you kind of brought it into visual,
Hmm.
like a drawing. Um,
Yep.
there's one, there's one where like I haven't gotten to that passage yet, but I remember from last time studying for the MCAT, like the Jack2 phosphorylates this and all that crap.
Mhm.
I feel like that's all another situation where like drawing out what phosphorylates and activates and inhibits
Yep.
would
Yeah.
be helpful.
What? Why don't you take a screenshot of this
Okay.
and then we can go over. I mean, yeah, if you want, we can like. Do you want to go over the questions here, or do you want to go to that passage with like the Jack? Oh, get all the stuff on the left too.
Oh, oops.
The legend, the
Oh, yeah. Oh, sorry. Yeah, what were you saying? Sorry.
Oh no, I was just asking what do you want to do?
Oh, I guess yeah. I guess we can go through this passage, and then you were saying what were you saying? You were saying something about Jack. Oh, you were saying the without the Jack Weston.
Oh no,
What?
sorry. I thought you mentioned some passage about like Jack 2 and stuff.
Oh yeah, yeah. That was just from my memory from taking it like a couple years ago when I was studying.
Got
I
it.
felt like that there was some passage like I haven't come across it yet, but there was like it was like a passage like Jack 2 phosphorylates this, and then it was like if you don't, well at least for me, I felt like if I don't write it out, which I didn't do for the actual exam, I feel like it would have been hard to kind of
Yep.
visualize it and stuff.
Yeah. Yeah, that's a another thing. Like I'm kind of surprised like how many students that I've had that are just so opposed to writing things down because of, the time crunch. And like I know there's time crunch and I know that, you don't have time to do a lot of stuff, but I always notice that I always would always be writing or drawing some stuff like stuff down. Not because another thing students say is like, what's the point of me writing it out if I'm not going to even use it? Um, but I think it's good that you draw and write stuff out because that's exactly what I do. And I I honestly like never, unless like it's like a passage that I can like really understand well from like sorry, unless it's like a passage where like the questions are relatively easy for me to answer, like I typically write things out or draw things out. Like so I think it's good that you do that.
Okay.
And
Uh,
what you showed me, like you do it also like you're probably like one of the first ones to like draw it out like the way that I do, like the way that you drew like your note and stuff, even with like the phosphorylation being like a P with a circle, like that's exactly what I do. So you're like very much on the right track. Mhm.
um, okay. I guess I guess maybe we can go through this passage and then
You mean like the questions?
yeah, I guess yeah. You think that'd be beneficial to go through the questions or um
You can
did
uh
you oh
yeah.
go ahead, sorry. Yeah.
I was gonna say you could hide the solutions as we
Okay.
go,
Okay.
but
Okay. Yeah. And do you ever do you think it's beneficial to do questions? I know you were saying to like do all the questions and then find out the ones that you don't understand. Is it ever beneficial to just do like a raw passage together? Is that waste of time?
oh, I mean, yeah, no, we could totally do that.
Okay, okay.
Especially if you know you understand these like the questions for these,
Hmm.
right? But yeah, I could we could do that. We could do anything really, whatever you want.
Um, I guess we can run through these passages together, and then I just want to quickly one thing that's been kind of bothering me for a while is Zeff, and then
Oh yeah.
something confusing me about electron questions. So maybe we can do that, and then if we have time, then we can just do like a raw passage together.
Yeah, do you have like an example of a question about Zeff or something? Like
I don't have like a
does
uh like a it was just from our I think it was from Jack Weston that maybe the content that we kind of did and that but I think that's where it came and I think we were kind of I was yeah,
uh-huh.
that's where it came from. I think it was a Jack Weston content question that kind of brought it up, like all the periodic trends. Most of them I understand how they kind of,
Yep.
but yeah,
Okay.
yeah.
So do you want me to go over like Zeff?
Yeah, I guess after this, after we get through like these five questions or whatever, then we can go
Okay.
through Zeph.
Got it. So
Yeah.
all right, this question, what bond is cleaved by IN during the first reaction of integration? So they are telling us that, yeah, that it catalyzes cleavage of a GT dinucleotide. Um, so what we're going to have here is, so I mean, we're breaking like DNA, right?
Hmm.
So they kind of made it nice for us with the choices all having phosphorus. Um,
So
because yeah.
you quickly kind of like like you quickly identified it already, like from the passage that line, right?
Oh no, that was more
Oh,
you were around there. I was looking up like you know up top. Um, but yeah, even if they didn't say that though, like we would still be able to figure it out because it says that it integrates viral DNA into the host chromosome, and the host chromosome meaning the host DNA. So you're taking viral DNA and you're inserting it in host DNA. How do you do that? Well, you're going to have to cut like host DNA, like when you had like your like the plasmid situation, like that's a different situation, but do you know what I mean, like by the plasmid, like kind of thing?
Yeah, like the bacterial conjugation and that kind of stuff, like with the viral or that's maybe that's bacteria, but
So,
the
like in bacteria, there's a circular DNA called a plasmid.
F plasmid or something. It's kind of it's there but rusty, but
Uh-huh. So, I think you're thinking about like conjugation with F stuff. But plasmids are just so it's like a way that we can make like insulin, like because prior to this, it was very hard to make like human insulin. So basically, like bacteria like this it has its own whatever DNA, let's just say it's that, but it also has this circular DNA called a plasmid. And what we can do is with a plasmid, we can cut it so we can have, something like this. And then we can take a gene for like insulin and we can insert it here so it can be fitted inside of that plasmid. And the plasmid's in the bacteria, and we let the bacteria, you know, multiply or, double itself, each time through like binary fission and stuff. So we end up getting a lot of bacteria that have the gene for insulin, um, and that's how we can make a lot of insulin. So this, like, just the mere act of adding DNA to DNA means you have to cut the original DNA so you can insert that DNA that you're trying to insert into it, right? Like, how could you, like, if you just think about maybe even the double helix, how can we add to this? We would have to cut it, right?
uh-huh.
So,
Yeah.
yeah, what were you saying?
Oh no, yeah, but yeah, yeah, cutting it like yeah, that makes sense. Yeah.
Yeah. So, um, yeah, I'll like maybe now explain it in like two ways. So one way, I'll start with a way that doesn't require this thing here, the cleavage of the GT dinucleotide, and then after I'll include that. So just very basically, conceptually, right? If you add something to your DNA, you would need to open up the DNA, right?
Yeah, like maybe helicase or something like that, or
So, so like the helicase and stuff can unravel like the two strands and effectively like, the hydrogen bonding between the nucleotides can also be broken by just, cleaving or sorry, separating the strands. But when you separate the strands, you can then have like a DNA polymerase like fill in the complementary strands. But if we are, and that's just taking like individual like nucleotides and adding them like one by one, right? But if we wanted to have like let's say this like blue viral DNA and we wanted to insert it in to the host DNA, we're gonna have to break some type of bond. And since the choices all deal with phosphorus, we ask ourselves, what type of, you know, so there's phosphorus in DNA. Where is the phosphorus in DNA?
Um, so where's the phosphorus in it? So the is it a phosphate backbone? No. Um,
Yes.
oh, okay.
Phosphate backbone, right?
Okay.
Because check, so some whatever base pairs like there, and then we have the um so the 3' OH looks like this. I mean, there's also like a phosphate here. I'm just going to draw it as a circle. But for this phosphate, I'm going to actually draw the phosphate. And whatever, some other nucleotide here. So you know how in DNA, like you need to have, well, in DNA replication, you need to have a free 3' end.
Yeah.
So that's what this is. So like if you count one, two, three, the OH, the hydroxyl on the three on this three third carbon here on
hmm
pentose is that free 3' end. And what do I always say about hydroxyl groups? When you see a hydroxyl group, it's the most common.
Most hydroxyl group, most common hydrogen bond, or no? No.
So remember this. Whenever you see, like, train yourself to, whenever you see a hydroxyl group, think nucleophile.
Okay. Okay.
And when you see a carbonyl group, that will be your most common electrophile.
Okay.
So here, I'm going to kind of show you an example here in green. So if we had a hydroxyl here and a carbonyl here, the carbonyl, since it has that electronegative oxygen, has a dipole making the electronegative oxygen have a partial negative charge, and the carbonyl carbon has a partial positive charge because the oxygen is pulling electron density towards itself, right?
hmm
And so the hydroxyl will nucleophilically attack the carbonyl carbon, momentarily pushing the double bond because carbon can only make four bonds, pushing this double bond to the oxygen, but it'll come back and it'll kick off the leaving group, and then you'll have the OH, bound to the carbonyl carbon. So this is something that you want to have down, and you want to train yourself to see that every time, you see a hydroxyl group or a carbonyl group. Now, similar to the carbonyl group is a phosphate group, right? Because the carbonyl group has a carbon that's double bonded to an oxygen, and the oxygen pulls electrons from it towards itself. The phosphate here has not only a double bonded oxygen, but three other oxygens. So it's having its electron density pulled and taken away from it by all these oxygens, thereby creating a partial positive charge. And so that means our hydroxyl attacks the phosphorus of the phosphate, pushing this up here momentarily, bringing it back and breaking it off from this. Well, actually, never mind that for a sec. It's just going to attach to this, and that's going to be adding the bottom nucleotide to the one up top in that backbone.
Okay. Okay. And phosphate. So the oxygen is more like electronegative than phosphate, right? So it's gonna have the pole, right? Is the pole gonna be towards the oxygen or the phosphorus? I was just looking at the periodic table now. Sorry, no, why am I doing that? Yeah, where would the pole be? Was it like in the phosphate group here?
Sorry, what I was just trying to look for a good picture, but what were you saying?
No, I just have the periodic table on my other page. So the oxygen, so this uh partial positive that you drew here is the pole is like the dipole moment or the pole it's going away from the phosphorus, right? Because the oxygens are more electronegative.
Oh, yeah, yeah, yeah. Yeah.
Okay, okay, okay.
So, uh,
And then, oh yeah, sorry.
uh, sorry, yep, go on.
And then, so this number three here, one, two, three hydroxyl. So this is DNA, right? So RNA
It,
uh,
yeah,
would have it would not. So I thought so deoxyribonucleic acid ribonucleic. So DNA does there's something DNA is deficient in something, right? Deoxy, like it doesn't have the difference between the RNA and the DNA.
yeah. So, good question. So basically, the RNA will have a hydroxyl group also on that second carbon,
Oh, okay.
but it's good that you brought this up because RNA is a temporary thing. It's like it's like your DNA is like your in I don't know, your like investment account that you can't really take out like money from or whatever, but the RNA is like a checking account where you put it in just to use it. So uh and so that means that RNA is temporary because it it doesn't stick around for two reasons. One reason is that in the cytosol there's a lot of um nucleases that break apart the RNA, but also it intrinsically is a less stable molecule because of that extra hydroxyl group, which can nucleophilically attack this carbon. So that can also happen, but yeah, that's what you're gonna see for the um RNA. So now I'm gonna share this image with you if you could pull that up. So this is exactly what happens. So you have on the left, you see that you have this like this is like a strand, right? And then this is the free three prime end. And then this is another nucleotide, like in the form of like TP. So it could be ATP, GTP, you know, um, those types of things. DNTPs, I think, is what they're called. And you see how, yeah, the hydroxyl attacks the phosphorus of the So also like counting the phosphates from the pentose, it's like this one is the alpha, this one's the beta, this one is the gamma. Now, there's two things that can happen in something like this. So in terms of DNA polymerization, we want this base, like, right, for DNA. So that's why the hydroxyl up here attacks the alpha phosphate because then these get cleaved out and we have that, which is what we want. And that's different from, let's say, another high yield concept, which is kinases. Do you remember stuff about kinases?
Yeah, so like kinases and phosphatases. Kinase kind of phosphorylates, phosphatase dephosphorylates.
So let's cover that again briefly because it's really high yield. So a phosphorylase. Let me see if I can do something here.
And I guess this will be helpful for glycolysis and all that stuff and gluconeogenesis.
Okay. So I'm going to just quickly. Okay. So I just connected my like Wacom pad. So let's see. Um, Actually, I'm gonna open up a whiteboard. Let me just do that real quick. Is it showing anything for you?
No, I think it stopped sharing on my end. Oh wait, oh Shane's whiteboard. I see now. Okay, I see. Yeah, yeah. Okay,
For some reason, it's still
yeah. I see your whiteboard now.
okay.
Mhm. Yeah.
So some reason, like the most typical, like just shows up as like blurry. Okay, got it. All right, so these are the enzyme classes. Did you see it, what I put in?
Mhm. Yeah, yeah, I can see it.
So you have your so now I'll add to this kinase. Now, what so a kinase is a transferase, okay? Now, this just transfers some functional group. It doesn't necessarily mean you're adding or removing. So in the case of kinase, it transfers a phosphate group. Now, that will be different from, let's say, a a phosphorylase, which adds a phosphate group, and also different from a phosphatase, which only removes a phosphate group. Okay?
Oh, okay. I never knew that. I always thought kinase phosphor because I thought about hexokinase, but okay, I didn't know that. Okay.
Yeah, so for instance, so here you could see that hexokinase is dephosphorylating an ATP and phosphorylating a glucose to give you glucose 6 phosphate and ADP. So that's exactly what you mentioned. But what about this?
Oh, I see. Okay. Pyruvate. Was that PEP carboxylate? No, it's pyruvate kinase. Oh, okay. Pyruvate kinase.
Now you see that we are removing a phosphate, right? From PEP.
Okay.
And because pyruvate doesn't have a phosphate, so we were going from phosphoenol pyruvate to pyruvate, we're losing a phosphate, right?
Okay. Okay.
Right?
Yeah, I'm a little rusty with the steps, but yeah, yeah, I think so, yeah.
What were you saying before that?
Sorry, I'm a little rusty with the steps, but I have to look at it again eventually.
Oh
So,
yeah, yeah. So yeah, this is just there's
so fine.
three irreversible steps.
Yeah.
These are two of them. There's just one more, which is just another um kinase phosphorylating something, but yeah, this one is going to be doing the opposite of what we think that it would do. So it would give the phosphate to ADP to make ATP. So you could see that kinases can add phosphates to substrates, or it can remove phosphates. Ultimately, it just transfers phosphates. Okay.
Okay.
Now, there's more to this because, like I said, it's very high yield. So there are three amino acids that kinases use to help them do their thing. Do you remember them?
Three amino acids that kinases,
Yep.
three amino acids that kinases help them do. You're talking about like in glycol, you're talking about like just in general? Or um oh, you're talking about the phosphorylatable amino acids?
Yep.
Oh, okay, okay. STY, I think it is.
So what are these?
Um, is it serine, threonine, and phenylalanine? I think so. Or is that NCQSTY? Yeah, I think serine, threonine, and I think Y is. I think it's phenylalanine.
Does phenylalanine like have
Or
so
maybe.
what do the serine and threonine have in common?
Oh, the OH groups.
All right. So what other amino acid can have that?
Okay, so then PFW are the aromatic ones. So Y is it tryptophan? No.
No.
Uh
So it'll be tyrosine,
Tyrosine. Okay, that's right. Tyrosine.
right? And so they all have what in common?
Oh, the phosphoryl phosphoryl the OH group that's be able to be phosphorylized.
Yes.
Okay.
So sure, phosphorylatable, but more so, I guess what I was asking was, yeah, that they have um hydroxyl groups, right? And what did we say about hydroxyl groups?
They are good hold on before I'm trying to decide between they're good so okay you said there's carbonyls and there's hydroxyl groups so they're good electrophiles. No, they're good nucleophiles.
Uh-huh.
And then carbonyls are good electrophiles.
Good.
Okay.
So, um, okay. So let's say we have um so this entire structure is ATP. I just didn't draw the ribose sugar and the adenine part, but you can see the three phosphate groups, right?
Yeah. Yeah.
And we call them like moving outward, alpha, beta, gamma. Okay. Now, the STY is going to have some OH group. And just like the carbonyl, carbon is the most common electrophile. A phosphorus in a phosphate should also be an electrophile, right? because it has highly electronegative oxygen pulling
Hmm.
electron density towards itself, right?
Yep. Yep.
So what will happen here is this will nucleophilically attack this gamma phosphate, which will, momentarily move this double bond up to the oxygen to make an O minus. But once it goes back to recreate the double bond, it'll kick this off as a leaving group, right? And then you'll have your OH attached to the phosphate. And, you know, and then you'll be left with ADP, which is exactly what I drew above, but, without the gamma.
And it wants to attack the gamma because there's less steric hindrance. Like, why won't it just attack the alpha?
Because
Right.
it just needs one phos phosphate group.
Oh, okay. Okay. Got it.
So yeah.
Okay.
So now that's so yeah, you want to know everything about that, the kinases. So that's pretty much for the kinases. Now, if we go back to this with DNA polymerization, for DNA polymerization, we want we don't just want a phosphate. We want a phosphate attached to that, ribose sugar and that base, right?
Hmm.
Like if we, yeah, so like that's why for this, we're not taking the gamma phosphate. Do you see where I'm writing stuff?
Yep. Yep. Yeah.
So here, if we wanted to, we could have just taken this gamma phosphate if we wanted a phosphate, but we want more than a phosphate. We want the phosphate attached to the actual nucleotide, right?
Hmm.
So that's why here it's a similar reaction, except we're attacking the alpha phosphate and we're breaking off these two over here. So you end up with this.
What happens to this? Oh, I could draw. Oh, what happens to this hydrogen right there?
Oh yeah, that's just this one. So here, let me use orange. So this over there is that,
Uh-huh. Okay. Okay.
right? And then this is just that, right?
Okay.
So yeah, you're gonna get this. I mean, it's also gonna be plus PPI, which is just inorganic or pyrophosphate or inorganic like diphosphate, aka just these two.
Okay. Okay.
Yep.
And this hydrogen just leaves? Like is that like just the one attached to the the oxygen?
Uh-huh.
Okay. Just kind of okay.
Yeah, yeah. So like that's something that I haven't really, whoops, drawn in depth. Like up here in this example, I kind of left this hydrogen, but it leaves because, you're making this additional bond. It's going to give it like a momentary like positive charge. And so something in the environment is going to want to remove that positively charged hydrogen. And so that's how that can happen, how that hydrogen can leave. So, okay. So we see that in DNA, we have what type of uh bond um regarding the type of phosphate.
Oh, sorry, what was that? What type of bond do we have? Or, oh, phosphor, would you say phosphodiester bond maybe? Or is that what you're asking?
Uh-huh.
Okay.
So, because we see that there's like, oh, whoops. So for this question here, it doesn't let me move it. Okay. You see I pasted it in the question.
Yep.
So what bond is cleaved by IN during the first reaction of integration? Now, we don't see any phosphorus bound to carbons, right?
Correct. Yep.
And we don't see any phosphorus bound to phosphorus, right?
Correct. Correct.
And we don't have any phosphorus bound to hydrogen, right?
Yeah, I mean that's what I picked. The wrong answer. That's what I picked.
But we do see that phosphorus makes bonds with oxygen, right?
Hmm.
So that must be the case. Here's the passage again. So, so okay. So remember I told you I was going to show you two ways. So this way, we can answer this question simply based on the fact that they said that that IN integrates, you know, the DNA into the host chromosome. And, if that means that we're adding that DNA to the host DNA. So the only type of phosphorus bond in DNA is the one that's bound or is phosphorus bound to oxygen. So that's one way of answering this. Now, the second way would be involving the thing that says cleavage of the GT dinucleotide. So if we had like GT, or rather, so the G is not complementary to the T, right?
No, it's to C, right? I think. Or yeah, cytosine.
The GT, because look, let's say, okay, let's say that the left side is the G, and there's hydrogen bonds with the right side, that's the C. We could unravel this strands to separate the G from the C, thereby, breaking those hydrogen bonds, right?
Hmm.
But they said it was a GT dinucleotide. So let's say this is T and this is A, and then this is the hydrogen bonds like that. So the GT dinucleotide is this, right?
Hmm. But they're not linked in any. They're not linked in any way, are they? They're not.
Uh-huh.
Oh, they are. Oh.
Yeah, they have to be, right?
Oh, I thought it was just G to C and then T to A.
So
Oh, okay.
those are linked together by,
Hydrogen bonds.
yeah, but the G is covalently attached to the vertical line, which is the phosphate backbone.
Oh, okay.
And so is the T, right?
Okay. Okay.
So see if I can find a picture to make it more clear.
The
Uh,
back.
okay. Uh, so for this, okay, so this image you see,
Uh-huh.
so
Uh,
we see that in the middle, like I'll use in the middle here, we have what we expect, which is T hydrogen bonding to A, G hydrogen bonding to C, etc., right?
yep. Yep.
And so essentially this means that so if we break these bonds, you would separate the strands, right?
Mm-hmm. Mm-hmm.
Okay. But now for like a GT dinucleotide, that would be this entire thing, right?
Yep.
So if we break that, what are we like breaking? At which part are we breaking this?
I mean, it looks like now you're breaking the hydrogen bonds and the covalent bonds both to do that.
Uh-huh. And so which one would be, because sure, we are breaking the hydrogen bonds, but you know, which one is going to be, I guess like that could be like a consequence, but if we're cleaving the GT dinucleotide, where are we like breaking a bond?
Oh, I guess we're the at the 3' end, like the third carbon and
So we want to cut there, right?
okay, okay,
Right?
okay. You want to retain the phosphate. Um, okay, okay. I was saying here, but I guess it makes sense
You're
to
saying
do.
where?
I was saying over here, but I think it. I guess you want to retain the.
There? Is that what you're saying?
No, I was saying here, but that doesn't make sense.
Um, yeah, I mean like that's still
Oh,
you're
okay.
still on the right track there. So let's see. This is I just wanted to show how it looks like in the So like, okay, so like this is and this, these are the backbones, right?
Hmm.
And
Hmm.
these are just the base pairs.
Okay. Base pair. Why is it called base pairs? I mean, I know it's called a base. I don't know. The nucleotide base pairs. Okay, got it, got it. I just okay.
Mhm.
Yeah. And it's negative backbone because of the phosphate groups, right? The carbon or sorry, the negative oxygen on the phosphate is why they call it negative
Yep. So this
nucleotide backbone.
picture that might help clarify things.
Okay, okay.
You see how on the left side we're doing a single-stranded break
Uh-huh.
like here. Oops. Like here between this T and this A, and that corresponds to that over there.
Uh-huh.
And what actually happens here is hydrolysis of the phosphodiester bond. And so that's what we're breaking when we, cleave like a GT dinucleotide. So for that question here, where they said the oh, cleavage of the cleavage of the GT dinucleotide. So that's another way that you could have figured out that we're doing a, you know, break of a phosphodiester bond.
Okay.
So this question could have been made harder if they had like as a choice like OH, right? Because people would then think that if you're breaking the hydrogen bonds between the bases, that could be what's happening. So, yeah.
Okay. Yeah.
Mhm.
Yeah.
Does that make sense?
Yeah. Yeah. The OH.
Okay.
Yeah.
For the next question, do you think you can screenshot it and then put it in this whiteboard?
Let's see. No, there's no. Ah, there we go. Okay, so then I just drag it from the chat or something. I put it in the chat, but is there a way to integrate it into the?
Yeah, you can copy paste it directly into the whiteboard.
Ah, okay. Nope, sorry, did I?
Were you able to?
Yeah, sorry, I've just never done this before. I'm pasting it, but it's not.
Yeah, I could maybe
The,
for you.
okay. Ah, okay, perfect. Okay, nice. Oops, sorry. Okay.
Okay. A vDNA sequence encoding a protein is inserted into a host genome by IN. The protein is translated from the hypothetical mRNA sequence shown. based on the passage, the segment of the original viral genome that encoded this protein had what nucleotide sequence? All right. Now, this, tell me your approach.
I was having like, first a bunch of things were going through my mind. I was like, okay, do I need to find like the stop codon? What exactly are they asking? And then I was like, do I need to find the complementary sequence? And then I was like, okay, maybe not the complementary sequence. It's just literally. I was thinking, okay, this is viral, so maybe it's just gonna be the exact same sequence as what's shown because we're not talking about like DNA. So I don't know, maybe not the best rationale, and that's why I chose A. It seemed to
So,
be, yeah.
so the first thing you said about stop, you said, wait, you said stop or start codon?
Stop codons.
Okay. So if they had, I mean, if they were asking us about the actual translation step of, amino acids being added, then sure, stop codons and things like that can affect that. Uh, but they're telling us that some type of vDNA sequence that encodes a protein is inserted in the host genome, and that the protein is translated from this hypothetical mRNA sequence. And, and then they're asking what was the original genome that encoded this protein? Like, what would that nucleotide combination be? So we're trying to find the original um, viral genome. Okay, so they're saying that we have this hypothetical mRNA sequence, and now we know that mRNA is from DNA in the nucleus. So if we were to write this as DNA, it would just be like CCGTT, something like that. Um, but since we're dealing with HIV, which has reverse transcriptase, we convert RNA into DNA, right?
Oh, okay. Yeah.
Okay. And we, so, okay. So we have our complementary RNA that will have, so, okay, let's see here. So, okay, so I think what can make this tricky is that they call it vDNA, but for, you know, a retrovirus, the genome is made of RNA, right? Because then you have your RNA being transcribed, reverse transcribed into the DNA. So, okay. So if they're saying that this hypothetical mRNA is the ultimate, thing that gets translated, we want to know what's the original, um, I guess the segment of the original viral genome that encoded this protein. And the so this can be the coding strand, and complementary to that would be the template strand, which would be like the three C, C, G, T, T, G, A. Oh wait, U, U, G, A, C, U, G, A, U. So kind of like, so it's kind of like this, but what is it exactly the same as that? Or, oh, sorry, that's with the, so it's like the CC. Okay, so let's say that this had, instead of the T's, the U's, it would look exactly like this, right? Or almost exactly.
Yes. Okay. Okay.
Now, it is it exact, does it exactly look like that? Or
So then this it looks like answer choice C is DNA, and then
is
what you wrote out was RNA, or because the uracil versus the.
I just mean like, like let's say that this was, that these had the U's.
Okay. Okay.
Now, is it what I wrote down here?
Yes. Yeah.
All right. So it looks exactly like it, except one thing about it makes it wrong or different.
So you did it complementary, right?
Mhm.
In the 3 to 5 direction.
Yep. So C is in the 5 to 3 direction, right?
Correct. Correct.
So yeah, so that can't work. So, but then from our template strand here, we can synthesize a new strand that's complementary to the template, which will just be exactly the same as the coding strand.
So the one you drew out, was that the template or was that the coding strand?
The
That
template.
the template strand. Okay.
Hmm.
Okay. No, when you talk about coding strand, that's talking about transcription.
So the coding strand is the one that actually has the sequence for like a pro to make a protein,
Oh, okay, okay, okay,
and complementary to that will be the template strand. But complementary to the template strand will be the new strand, which will be equivalent to the coding strand.
okay, okay.
So, yeah, I think that was a great guess that you made. 'Cause I remember when I was first doing this, I instantly eliminated um A and D because I saw vDNA here, and while I knew that HIV had reverse transcriptase, I figured that they would just at least not call it vDNA, at least call it vRNA or something. But yeah, that's just what I personally thought was difficult about this. Um, but it seemed like you knew to use the RNA, and yep, so it's just going to be exactly the coding strand.
Okay. I always just feel like AMC is trying to trick you. I mean, I don't know if that's a good mindset to have, but
Well, yeah. There's going to be a lot of that. Like, rarely will it be like so straightforward.
okay.
So the next question,
Oh yeah.
like you could instead of because we already have the passage, you could just screenshot the question and add that in.
Okay. Sorry, I don't know. I put in the chat for whatever reason when I try to paste it, it doesn't allow me to.
Okay, I got it.
Sorry.
Okay. So it seems like you got this right also. Do you
This
understand?
one- yeah, this one I felt good about. I think this is just kind of like, do you understand like research and like independent and dependent variable and like control and negative, that kind of stuff?
So like what we're having here is just we have to notice or recognize that so IN is the enzyme and the substrate is the vDNA and the ODN they said was a competitive inhibitor that looked like the vDNA. So that's going to be what the ODN is in the presence of and absence of the ODN. So this would just look like something like, well, when we typically have Michaelis-Menten, we have like our substrate concentration here, our V here, and something like that, right?
Hmm.
So, or we have something like this with like no inhibitor, and then with inhibitor could, lower it like that or something. So we're doing the same thing, except the substrate here is going to be your vDNA, and your IN concentration is your enzyme concentration, which the Michaelis-Menten assumption uh states like should be um constant. And then yeah, it would be like, the presence or the absence of ODN and the presence of ODN. So pretty much that.
I haven't I need to like refresh it, but like, did you have like a good way to like um is it just essential just I mean would you is it essential just memorization like I have to refresh myself on the plots like the Longweaver and Michaelis-Menten plots and just all of the maxes and KMs and how they differ like competitive non-competitive would you recommend just kind of straight up memorizing it or
You're asking me the effect of inhibitors, like
yeah like all those plots like just straight up memorize that stuff or what?
yep.
Okay, okay.
Yeah. So like yeah. So memorize the effect it'll have on KM, if any, and the effect on Vmax, if any. And I guess it can help to think of perhaps, whoops,
Oh, sorry.
perhaps like let's see. So all inhibitors lower Vmax except for competitive inhibitors. Okay. And and I got or okay, maybe I'll do okay, so maybe I can make like a table. So for instance, competitive, uncompetitive, non-competitive, or here, let me just put in a image here. Okay. So in this, yeah, so I would memorize this chart. So competitive inhibitor raises Km, has no effect on Vmax, and it only binds to the enzyme. And for the uncompetitive inhibitor, it binds only to the enzyme substrate complex, and it lowers the Km, and it lowers Vmax as well. And your non-competitive inhibitor can bind to the enzyme and the enzyme substrate complex with equal affinity, has no effect on Km, but lowers Vmax. And then you can have a mixed one, which you won't see too much of, but it'll be able to bind to the enzyme and enzyme substrate complex not equally. And its effect on Km uh, can go up or down, but its Vmax will always go down. And then next to it, I put um,
Hmm.
the Lineweaver-Burke plot.
Yeah,
So,
that yeah, I guess I always have trouble like this stuff I can, get down pretty quickly. But like conceptually I have trouble understanding like for example, why mix inhibitor can be up or down. And then like I don't even understand. I still don't like I understand like okay, people would say okay, Vmax is one half the Km, but it doesn't mean anything to me. Like I'm trying to understand
mhm.
the it's like okay, it's people say it's the concentration at which the substrate is half the velocity or something, but it doesn't like conceptually like stick to me. I don't understand what they're talking about.
Got it. So a lot of times we use values that are at the halfway mark just to help us like describe things. So like for instance like half-life, right? Time it takes for 50% of something, to decay, right?
Hmm.
What about LD50?
LD, oh, so it would be 25 and then 12.5 and then
Oh no. So LD50 is lethal dose 50.
oh, 50. Okay.
So it's the dose that will kill 50% of the population. Like if it was some drug or poison or something.
Hmm.
You can also have you also have TM. Whoops.
Is that melting point or something?
Yeah, it's the
Oh.
temperature at which 50% of DNA or a protein, gets whatever, let's say denatured or messed up or, melts. So 50% of something is just something we like to do to help us conceptualize things. And in the context of Michaelis-Menten, where you typically have some asymptote for Vmax, And, you know, you start off with a direct proportion, meaning higher amount of substrate means a higher amount of V, which is the rate of the reaction. So it always starts off linear like that, right? Now, the only reason it doesn't stay linear is because of the Vmax asymptote. So then it starts to go like that. So what's the point of, you know, Km being the substrate concentration at 1/2 Vmax? It's so that we have a it's so that we have a measure of the initial rate of the enzyme before it starts to, you know, plateau. So this could be 1/2 Vmax, and then this would be Km. But given the nature of uh, enzyme kinetics, you know, it'll initially have a linear increase and then it'll just, you know, plateau. So if you didn't have Km, like, you know, you would either have something like beyond, like something up here, which doesn't tell us anything, or something like way before uh you get to see anything. So the Km that is just there for us to be able to get a good idea of the enzymes, you know, of the substrate concentration needed for the enzyme to be like showing in a linear kind of relationship before it plateaus.
So if there's okay, so if there's fewer substrates available, does it become like more like exclusive in the sense of like the affinity like the affinity becomes higher because like it wants to there's less available, like the Km affinity that
So let's see. So I mean, so we see from this graph that if we increase substrate concentration, we increase V, right?
yeah. Hmm.
Um, and then that the Km is the substrate concentration at 1/2 Vmax. And so, and then you're talking about affinity. So if the Km is small, it means that, or like, you know, comparatively, like let's say that we had a different Km here, like to the left,
Hmm.
that Km would enable the reaction to be at 1/2 of its maximum rate with a smaller amount of substrate. So if Km decreases, the affinity increases. So I'll write that down. I'll say Km is inversely proportional to the affinity. So, and that's true for these other things that I talked about on the left. Like if the LD50 is the dose that kills 50% of the population, if you had a lower LD, if there was a lower LD50, it means that there's a higher, well, it won't be affinity in this case, but like a higher lethality because you need less to do that same amount, right?
Okay, so you wouldn't say okay, LD25 wouldn't make sense. You would just say a lower LD50, right?
Yeah, so comparing two different LD50s, if one is like 5 and, you know, let's say it's like LD50, 1 cup of coffee, if, you know, coffee kills you. And from there, we went to zero point 0.5 cups of coffee. It means that, well, if the LD50 is zero point 0.5 cups of coffee, that coffee is much more lethal. And then same thing with TM, right? if that's the temperature at which 50% of the DNA or the protein get, denatured, if let's say TM is 100 degrees, let's say another TM's 50 degrees, the lower TM means that you don't need to raise the temperature as high to get 50% of the thing to, denature. So that's just something that we use, uh, you know, as like kind of like a standard for helping us understand these things. So,
Okay.
yeah, that answer your question about the.
Yeah, it's making more sense now. Yeah. Like it's
So like, so if we were to see, so now if I was to do this, if we had a competitive inhibitor, what would this look like?
okay. So a competitive inhibitor. So it's going to attack at the active site, not the allosteric site. So it's going to attack at the active site. Um, so it's going to compete for so it's going to compete for the ends for the substrates. Um,
So think about it in terms of what effect does it have on the rate and what effect does it have on Km?
um, okay, so on the rate, I don't think it will affect the rate because then you just have more you because you just have more substrates, put out more substrates to overcome any sort of variation in rate. Oh, okay. Effect on Km. So it increases Km.
So it increases Km, but it does not change Vmax, right?
Like conceptually, why does that happen? Why does it increase the Km?
So we know from here, whoops, Km is inversely proportional to affinity, right? So
Hmm.
if you have an increase in Km, the affinity goes down. Now, the um, enzyme, let's say this is the enzyme, oops, and let's say this is the inhibitor, and let's say this is the substrate. Um, if the inhibitor, and then this is the active site of the enzyme, if the inhibitor binds to the enzyme, the substrate won't be able to bind to it as easily, right?
Okay. I had a reverse in my mind. I had the substrate. I don't know why I always had the enzyme attacking the substrate. Okay, so it's good to know that you reviewed that. I had it backwards.
Oh, were you saying that you were thinking the substrate would attach to the enzyme?
Yeah, like as instead of the enzyme, it would be like the large one. The substrate is like the. But okay, so I just had to review that. So
Oh,
eh,
so yeah, the enzyme is going to be the bigger thing. The
okay,
substrate is the thing that it takes in, and the product is what it you know, and
Okay.
turning the substrate into.
Got
But
it.
for this, the competitive inhibition means that the substrate must compete with the enzyme, which means that you're going to need more substrate to be able to compete with that competitive inhibitor. So, like theoretically, you could actually overcome the effects of a competitive inhibitor by adding substrate. So, so that means that Km, if Km increases the affinity, so this means that, okay, so must compete with inhibitor because inhibitor has higher affinity than the substrate does.
Okay. Okay.
So that's why so now since Km is inversely proportional to affinity, in order for uh so that means that a competitive inhibitor would result in an increase in Km, which means a lower affinity for that substrate for the enzyme.
Okay.
Right. So that's why Km goes up. So that would look like something like this. So competitive inhibitor. So we can see that the Km has shifted to the right, meaning we're increasing the Km, but the Vmax, so maybe it's a little, oops, hard to see here, so I'm going to make it a little bit more like that. So you could see that so for the competitive inhibitor, the Km increases, which is why it shifts to the right, but the Vmax stays the same, right?
Yeah. Yeah, I see that now. Yeah.
Good. So, and for non-competitive, or I guess we could do uncompetitive. So the uncompetitive binds to the enzyme substrate complex. So up here, I can kind of draw that again. So here we have our enzyme, and we could have our substrate attached to that enzyme, and then we can have our inhibitor attach to an allosteric site, which I'm going to place down there.
Okay.
So, and then this causes a decrease in um, a decrease in Km. So that's just going to move to the left a bit, but it's also going to decrease Vmax. So this would look like something like this, but it'll end up plateauing lower. And that's your uncompetitive inhibitor. So since the Km is going down, it's a leftward shift, and the Vmax is also going down, so the overall thing goes down. And then lastly, for the non-competitive, I'll use dark purple. The non-competitive has no effect on Km, um, but has an effect of lowering the Vmax. Right. So that'll just look like that. So it's kind of harder to see in this way, so that's why they typically will just show you it as a Lineweaver Burke plot like this.
Mhm.
It's much easier to see. But but yeah, those are the types of inhibitors.
Okay.
Yep. So any questions there?
No, I think I just need to review those. Those are
Okay.
high yield. No, I said yeah, no questions. It's making more sense now to me. Last time around, I just kind of memorized it and didn't really
Mm-hmm.
fully understand it. So
Got it.
yeah.
So from the passage, so you understood this question. So what about the one after 21?
One, okay. Let's see here. Just
Oh, yeah. So we knew this was going to be something that they asked because they love to ask this type of thing, this type of question. So expect to see this type whenever you see them talking about, the amino acids, which will be all the time. So we know the amino acids at the active site are negatively charged. So if you replace it with a different, so they're saying position 64, and that's according to the passage, aspartic acid, right?
64D, yeah, D64. Yeah.
So if we replaced it with asparagine, that doesn't have a charge, right?
Asparagine. Um,
Because the only ones with charges are the acidic and basic ones, right?
correct. The rest are, yeah.
So the rest could be either partial charge for polar or nothing for non-polar. So if they replaced it with asparagine, which has no charge, that should mess it up, right?
Hmm.
And if we add lysine, that's going to have a positive charge. So that's going to mess it up too.
Uh-huh.
And then valine has no charge, so that's going to mess it up.
Oh, okay.
So
Okay.
the only thing that will maintain the function would be another negatively charged one, like glutamate or glutamic acid.
Got it.
So this type of question they'll ask pretty much all the time, so
Mhm.
expect it. And then the next one,
I think. Oh, um, let's see. Let me, I didn't get to that one, but let me, um, Does it come up or I don't know if it came up or not.
Okay, got it. Oh yeah, so see, this is also what we were expecting, right?
Hmm.
So that's the buzzword thing, right? When we saw 288 residues, that's a buzzword that signals, hey, they're probably going to ask you for the weight of it. And then when they mentioned dimers and tetramers, I was also saying that they're probably going to ask you the weight of that, right?
Yeah, yeah.
So, okay.
So, so you said,
Yeah.
um, okay. I think so you just, was it 110 Daltons and then,
So yeah, the average weight of one amino acid is 110 Daltons. Um, so this question's asking about the tetramer, right? So how many residues would a tetramer have? So one IN has 288. I'm just gonna write 300 for one. And for a tetramer, it'd be four times that, right?
hmm.
That'll be around 1200 amino acids, right?
Uh-huh.
And then I could use dimensional analysis. So for one amino acid, it's 110 Daltons, which I can write as um, zero point 0.1 kilodaltons. Does it make sense how I did that?
Yeah, you just three because kilo th three. So um
So now 1 over zero point 0.1 is the same thing as 1 over 10, which is just dividing the 1200 by 10. So I get 120 kilodaltons, right?
uh how'd you do that last part again? Sorry, I guess that's a good trick to know. So you said, oh,
Oh, yeah.
point.
I always like fractions. So zero point 0.1 is 1 over 10.
Oh,
So
okay, okay, got it.
yeah, I always favor using fractions. So yeah, we get 120, and this is our closest one. Why do you think this is a little larger than what we got?
Um, so it was 288, and that was in the numerator.
Uh,
Oh,
so we rounded the 288 up.
yeah. So is that what caused maybe the final answer to come lower? I don't know. Maybe that was my best guess. So I don't know.
Yeah, so we rounded the 288 up, which should have given us a larger number, but we
Uh-huh.
but we ended up with a smaller number. And that's because the 110 Daltons I just treated as 100
Oh, you did. Okay. Okay.
here.
Okay. Got it. Got it.
Putting 0.11, I just
Okay.
put zero point 0.1.
Okay.
Um so but that could have so I also have the fraction value of that memorized. So 0.11 is 1 over 9. So 1200 divided by 9, I could say 3 goes into 9 three times, 3 goes into 12 four times. 400 divided by 3, 3 goes into 4 once. With the 1 left over, 3 goes into that 3 times. Then there's 1 left over, and 3 goes into that 3 times as well. So 133-ish. So it'll have to be between 120 and 133. So that's another way of doing it, figuring out a lower bound, higher bound. So that makes sense.
Yeah, that makes sense.
Okay. Now, I think you said that you didn't do this the rest of this, right?
Yeah, I didn't do the rest of this.
So I'll let you do the rest of that.
Okay.
And then I'll quickly go over Zeff.
Okay.
Yeah, mention that.
Yep.
So, like, okay, so we have in an atom, we have, let's see, so, okay, so our nucleus is composed of what?
Um, Protons, neutrons, and electrons.
In the nucleus?
Oh, and the nucleus is just protons and
And,
neutrons.
and neutrons, yeah.
Okay, and then electrons are on the the outer shells.
Yep, yep. So there should be a attraction between the positively charged nucleus and the negatively charged electrons, right?
Mhm.
So, for instance, if we had, um, let's say we had one, two, oops, three protons, okay? And then let's say we have, um, what's that? So the Zeff is Z minus S, and what Z is the number of protons, and what the S is number of core electrons, the ones that are, closest um, to the nucleus. So for this, and let's say we have an electron there, an electron here, um, we would have a Zeff of three protons minus two electrons to give us a positive one Zeff, right?
Mhm.
And if we had, please blue, if we had, let's see, okay, let's add another proton here, and then we'd have 4 minus 2 or plus 2 Zeff, right?
Mhm.
So that means that, or let me do this also. So I'm gonna put in just so I can make this like a actual element. Um, oops, another electron here. So that would give us, oops, that would give us lithium. Um, I didn't draw any of the neutrons, but lithium would have three protons, two of like the core electrons, but three electrons total. So that's for lithium. And then for this one, So that's when I added um, another proton. And so I'm going to also add another electron out here. And so this is going to be like beryllium because beryllium. So what I'm trying to show here is that if we, from like the periodic table, which is like this, and we have like, I think like lithium, and I think like beryllium is here or something. Let me quickly do. So if we look at our periodic table, we could, oh, beryllium is right next to it. So we could see that for lithium, we have our Zeff of +1, beryllium, a Zeff of +2. And you can see kind of like a trend here that if we move from left to right, we are, and okay, let's say we did boron over here. So boron has 5 protons and it'll still have two of the core electrons because it'll be 1s2, 2s2, and then this one over here. So let's see.
The image is not loading, but I have the periodic table pulled up here. I don't know why it's not loading. It's weird.
Oh yeah, it does that. Um, whoops.
Okay. Yeah, that's better.
Yeah, no?
Yeah, I see it now.
Okay. So the oh yeah, so for the boron, we have 5 protons, and we still have the two inner two core electrons. So the Zeff would be 3 +3, right?
And you got you calculate two core based off of uh
The inner shell.
uh okay.
Like the innermost shell.
How do can I get that can I extrapolate that data from the periodic table or like
Yeah, yeah. So that's
huh.
what I'm getting at here. So we can see that if we move from the left to the right on the periodic table, we are
Uh-huh.
we're adding both a proton and an electron.
Okay.
And since Zeff is dependent primarily on the core electrons, the electrons of the ground state, the first energy shell, right? Um, the Zeff is going to increase left to right on the periodic table because we are adding protons and we're not really, I mean, we're adding electrons, but they're in the valence shell. They're not in the ground state shell, the closest shell. So that's why Zeff goes up from left to right.
Okay.
So, um, yeah, what do you think about that?
Um So Zeff goes up from left to right because you're increasing the number of protons, but you're also increasing the number of valence electrons, not so much the core electrons.
Correct.
And
Mhm.
and and then so I guess what helps me understand then is like the difference between Zeff and ionization energy. And then what's confusing me is like electronic repulsion, or does that have nothing to do with any of these trends? Electronic repulsion.
Oh no, it does. It does. So
Okay.
that can contribute to something called shielding. Okay.
Okay.
Uh so what that means is um so there's shielding caused by neutrons. And so okay, let's see if I can bring this all together. So basically all the periodic trends comes out of how tightly um a valence electron is held. So, for instance, so yeah, left to right, you're increasing Zeff, but if you go down, you're increasing the valence shell, and you're also increasing the distance of that valence electron from the nucleus of that atom. And so that Zeff, which is if we think about it as pulling the valence electrons, right?